Good morning, and thank you for that introduction, and I hope that the meeting went well yesterday. Thank you also for the opportunity to contribute to the meeting and to share some of our experiences in biomarker discovery and in developing diagnostics from both an academic and commercial perspective. For many reasons, I'm very disappointed that I can't be there in person, but I hope that this video presentation will be informative and provide a brief update of some of the work that we're currently engaged with at both the University of Queensland and with INOVIQ. It is axiomatic that understanding the processes from discovery to diagnosis is critical to improving diagnostic applications and patient management and outcomes. It is not necessarily, though, a linear pathway. And in today's presentation, I want to talk with you about the process of delivering new diagnostics to market and to share with you some of our learnings on how to improve efficiency and optimize the process. To do that, I will draw from both our academic and commercial work in the discovery of extracellular vesicle-associated biomarkers and of EV-based diagnostics. The most important concept that I want to communicate today is that if your goal is to identify clinically relevant biomarkers for the development or improvement of diagnostics for any clinical condition, it is to think and plan from diagnosis to discovery, not the other way around. What do I mean by this? It means that having a clear and precise definition of the intended use of the diagnostic to be delivered and how it will be implemented in a routine clinical pathology laboratory or as a point of care device before commencing the project is the most efficient and cost-effective way to ensure a successful outcome. To put that simply, and drawing upon the words of the late Stephen Covey, and from one of the more impactful and influential books that I read during my early scientific career, begin with the end in mind. It's really an axiom that not only applies to developing diagnostics, but to all aspects of human endeavour. If we have a clear understanding of what contributes to the final diagnostic test and what clinical pathology instrument it will be run on and what regulatory pathway it will be certified, many of the challenges associated with translating research into clinically actionable diagnostics are minimized. We are able to make more informed decisions that align with that process, including the selection of sample processing and biomarker identifications that best align with the final assay format, the equipment and methodologies for quantification of biomarkers, data storage and analysis, and the type of quality management system that the entire project should be run under. The result is the generation of regulator-ready data packages for submission to regulatory agents such as the FDA as a 510(k), a PMA or a de novo application. By so doing, the process becomes a continuum from concept to diagnostic application between and across academia and industry. But before we go any further, I should declare my relevant interests. I'm currently employed as the Chief Scientific Officer of an Australian ASX-listed biotechnology company called INOVIQ. The mission of the company is to develop and commercialize extracellular vesicle precision diagnostics and therapeutics for cancer and other clinical conditions. The company has developed a suite of tools for isolating subpopulation of extracellular vesicles for general use or for specific applications, including oncology and neurodegenerative conditions. And using these tools, the company is working with both academia and industry to develop and deliver EV-based diagnostics and engineered EV-based therapeutics. And I do refer to these products during my presentation. There are only two things that I want to focus on today. Firstly, and in relation to the concept of beginning with the end in mind, I want to present a few ideas about design control and why it's so important for biomarker discovery and the delivery of diagnostics to market. And secondly, to consider some of the challenges and solutions in identifying clinically relevant biomarkers and translating them into clinically actionable diagnostics. So what do we mean by design control? Design control refers to a systematic process that ensures medical devices, including in vitro diagnostics, meet user needs, intended use, and specified requirements. The FDA has provided guidance for the design and control of medical devices, including in vitro diagnostics. And it involves a series of well-defined steps through the planning and development process, including the identification of user needs, the clinical gap, how the test needs to perform to meet that clinical gap, that's the design inputs, the development of the prototype, the design process, the performance of those prototypes, the design output, is then checked against the design input in an iterative process. Once the design outputs have been verified against the design inputs, then the medical device is validated against the user needs, i.e., does the in vitro diagnostic satisfy the clinical gap, satisfy the user need. Perhaps the best way to illustrate this is using an hypothetical in vitro diagnostic. So on the left is the Symphony test. It's an FDA approved IVD now. Its intended use, the user need, was as a first trimester risk assessment test for complications of pregnancy. So the process of building this in vitro diagnostic was first to define the performance of this test that would satisfy the clinical need. And here is a design input table for this particular diagnostic. And what it does, it defines everything about the test that we need to satisfy to make it to work. In this case, it's using a plasma sample. It's a multivariate index assay looking at extracellular vesicle-associated peptides. We don't want to be measuring more than 10 of those. We need specifications, performance specifications for sensitivity and specificity to be high, high positive and negative predictive values. How is a test going to be delivered in a clinical pathology laboratory? Well, in this case, it's being delivered as a multiple reaction monitoring test on a liquid chromatography mass spectrometry as a class 2 in vitro diagnostic. We don't want to use more than 500 microliters of plasma. We're going to isolate those extracellular vesicles using a fully automated process that can only take less than 2 minutes per sample, and it's got to have really tight coefficients of variation, say 5%. Given it's an early pregnancy test, we want to be running probably 500 plus samples a day. And initially, we're going to implement it in a clear laboratory as a laboratory developed test so that we can develop real world clinical data, and the whole process will fall under the regulatory standard ISO 13485. So these are the criteria that we set before we do any work that we need to satisfy to meet this clinical need. So the next step in delivering this diagnostic is to build the prototype and evaluate its performance. And its performance is then designated as the design outputs. And here we can see the design outputs the performance of the Symphony test prototype that we've developed. And we can confirm that we're using plasma. We only need 5 biomarkers to develop an effective algorithm to deliver appropriate sensitivities. It works on the LCMS. We're using 300 microliters, not 500, so that's an improvement. On a fully automated platform, we're taking an average of less than 1.5 minutes per sample, and we have suitable coefficients of variation. With that particular platform, robotic EV isolation platform, we can isolate 12,000 samples a day. It can be implemented in a clear lab, and we have kept appropriate records such that the whole work will conform with ISO 13485. So the output design performance criteria are satisfied. They meet the design inputs, so the process has satisfied verification. Now the medical device can be validated against the user needs, asking the question, have we actually satisfied the criteria to deliver a first trimester risk assessment test for complications of pregnancy that performs with suitable diagnostic performance. And the whole process can be graphically summarized here into the discovery phase, the validation qualification phase, and commercialization phase. And this whole process is conducted within the framework of an appropriate regulatory standard, in this case, ISO 13485. And it's this information, the data obtained in the discovery, validation, and qualification phase that is used to generate the regulatory certification application. And in this case, it was a 510K application. And just to show you what a real 510K decision summary looks like, this is the one for the Roche Alexis CA1532 assay. And the type of information it details is the class of in vitro diagnostic, in this case a class 2, the intended use of the diagnostic, and it's noteworthy to look at the detail here. It's an immunological in vitro assay for quantitative determination of CA15-3 in human serum and lithium heparin serum to aid in the management of breast cancer patients. In conjunction with other clinical and diagnostic procedures, serial testing with this assay may be an aid in the early detection of recurrence in previously treated stage 2 and 3 breast cancer patients, for monitoring response to therapy in metastatic breast cancer patients. So it's fairly detailed, and this, remember, is where we start with the development of the in vitro diagnostic. In addition, it identifies the instrument to be used and the predicate device that the 510K was established on. It provides quite detailed descriptions of the test principles and methods. In addition, there's detailed reporting on analytical validation parameters, analytical specificity, and the clinical studies that have been conducted to validate its clinical efficacy, clinical cutoff values, expected ranges, etc. The FDA provides a document for the industry in terms of what is required for a 510K, and I would refer you to that document. The actual documentation for a 510K application is quite extensive. Here's our design control documents down here, and most of the files form the design history file, which is the auditable documentation of the entire development of the in vitro diagnostic and its implementation. To summarize the key points in design control, For effective development of in vitro diagnostics, we suggest beginning with a clear understanding of the regulatory requirements for your IVD, be that a 510K, a PMA, or a de novo application. This will inform many of the decisions around the selection of sample processing methods, analytical methods, and indeed the way the in vitro diagnostic assay will be implemented. It will mitigate many of the obstacles around the translation of research into clinical applications. For a seamless process, work under a suitable quality management system or regulatory guideline, such as the FDA's 21 CFR Part 8020 or ISO 13485 for medical device development. Implement a rigorous design control process and initiate and maintain that design history file as soon as possible in the process of research and development. Why would you make the time and effort as an academic researcher or a clinical researcher to go through that process? One, it ensures quality and reproducibility of the data generated, and it's the generation of regulatory ready data, a data package that can be submitted to a regulatory agency for review and hopefully approval. It adds value to the work that is being conducted, and I guess ultimately when it goes wrong, it goes very wrong. Not to dwell on it, but for example, this is the Amgen experience where they were evaluating candidate drugs from papers published in academic literature, and after 6 months effort they found that it was really a waste of money and time. They couldn't reproduce the results. Glenn Begley and colleagues went on to look at the reproducibility of preclinical oncology data and reported around 90% of it was not reproducible. Subsequently, Friedman and colleagues did a meta-analysis on the economics of reproducibility in clinical research and concluded that it was costing in the US around $28 billion per year, something to be avoided and can be simply avoided by implementing the appropriate design control and quality management systems. Okay, so let's move on to the second part of the talk, extracellular vesicle-based diagnostics. This is an area that we have been working on for many years, trying to resolve some of the significant challenges in translating extracellular vesicles into diagnostic applications. And really there are two major challenges. One relates to the heterogeneity of extracellular vesicles and reliable, consistent methods for their isolation. And the second is scalability. To deliver an EV-based diagnostic, you need scale. You need to be able to process 500 plus samples a day, depending on the in vitro diagnostic being used. And many of the methods that have been used in discovery are not suitable for that type of application. So let's segue into this section with this video from Caris Life Science. It's a fairly old video, but it's quite useful. EVs can be generated obviously by a number of different processes, one involving the invagination of multivesicular bodies, selectively or non-selectively capturing signaling molecules from the cytoplasm, fusing with the plasma membrane and then releasing them through an exocytosis process. EVs can also be formed by the outward budding of microvesicles or ectosomes from the plasma membrane, again containing signaling molecules that are released into the extracellular space. Once in the extracellular space, they can find their way to proximal and distal tissues, either fuse with the plasma membrane, activate cells by receptor-activated mechanisms, be endocytosed to deliver messages to cells, and we believe that they regulate cell function. And a number of the advantages of extracellular vesicles is that they protect those messages from degradation in the circulation, and they inform us about the status of the cell of origin. The video, while useful, does present a simplistic view of extracellular vesicles, and much has changed since its production. Not only do we now need to consider extracellular vesicles, but also extracellular particles, of which extracellular vesicles are a subset. Extracellular particles are non-membrane bound particles that are released from cells and have biological function. Within membrane-bound particles, we've identified both replicating and non-replicating particles that may co-isolate in our preparations and have different biogenic origins and modes of release. So in terms of trying to identify your favorite extracellular vesicle or exosome, we now have to consider the possibility that many of these particles are co-isolating and presenting challenges in terms of biomarker discovery. The potential issue of co-isolation of extracellular particles in preparations of extracellular vesicles was further highlighted by a recent review by Jepsen et al. In 2023. They were working with a number of extracellular particles and looking at their isolation using different methodologies. And if we plot diameter against particle density, what we see is methods such as sedimentation and precipitation result in potentially the most heterogeneous populations of extracellular particles. If we look at size exclusion chromatography isolates particles on the basis of their hydrodynamic diameter, and not selective on charge or density. Even density gradient centrifugation, which was considered gold standard, isolates particles of similar density but different size. And none of these isolate extracellular vesicles per se. They're isolating particles based on their physicochemical properties. The other possibility is to isolate particles based on their surface epitopes. So specifically targeting protein or glycoprotein epitopes on the surface of vesicles, not relying on their physicochemical properties as the mode of isolation. We have confirmed these observations in-house, where we have isolated extracellular vesicles from the same plasma sample using size exclusion chromatography, precipitation, or various immunoaffinity isolation procedures. And what we find is that there is a great variation between the recovery of messenger RNA and microRNA dependent on the isolation technique used. And these observations are consistent with what we see in the literature. Many studies completed to date have reported heterogeneity of EV preparations that makes data interpretation and comparative analysis challenging. And indeed, in terms of developing EV-based diagnostics and therapeutics, it would be an advantage if we had approaches that targeted discrete, well-defined subpopulations of extracellular vesicles. In 2022, a group of us prepared a scientific statement for the Endocrine Society on the emerging role of extracellular vesicles as cellular messengers. And in that statement, we identified the same principal issues facing the field, that of nanoparticle heterogeneity, the types of isolation procedures used, the lack of subpopulation analysis, and the challenges around capacity and scale to deliver extracellular-based diagnostics. Again, we reviewed the available EV isolation methods and scored them on a number of criteria, including the time taken per sample, cost per sample, whether the technique was scalable or not, the recovery and the specificity. And particularly noted that immunoaffinity capture-based methods performed extremely well, relevant to the development of EV-based diagnostics, scoring highly in the scalability and specificity criteria. So in terms of user needs for EV isolation, we identified requirements for rapid, simple, cost-effective methods compatible with downstream analysis, ones that enrich clinically relevant subpopulations of extracellular vesicles, that's reproducible, scalable, fully automatable to allow the capacity, and compatible with pathology workflow laboratories. The design inputs for a fit-for-purpose EV isolation tool may therefore be that it was rapid, requiring less than 2 minutes to process an average sample, of high throughput, cost competitive, reproducible, requiring low sample volume, around 500 microliters of plasma and 2 ml of cell-conditioned media, for example, delivered a total protein yield of 10 microgram per sample or more, a total RNA yield of 3 nanograms or more, and was scalable, fully automatable, and compatible with existing commercially available isolation kits. Based on these input design criteria, we have developed a paramagnetic immunoaffinity capture bead for isolating extracellular vesicles. The bead targets 9 different epitopes on the surface of extracellular vesicles and is designed for either on-bead analysis or on-bead lysis and downstream analysis. Using this bead in plasma, we're able to identify canonical EV proteins such as CD63, CD81, CD9, TSG101, and flotilin. We have also compared it to 4 available commercial kits. The ExoNet product here compared to the other kits for the isolation of microRNA, looking at CT values, and here for messenger RNA, again ExoNet shown in yellow. Working with life sciences company Promega, we have now fully automated EV isolation. The process is available for medium throughput on Promega's Maxwell platform or high throughput 96-well plate processing on Thermo Fisher's Kingfisher platform. The lysates from either platform are suitable for protein and RNA analysis. In terms of the design outputs, the performance of the fully automated EV isolation system, we are seeing that the design outputs satisfy the design inputs in that the average sample processing time is less than 2 minutes. We have capacity of more than 500 samples per day, and it will operate on low volumes of plasma and cell-conditioned medium, delivering total protein yields and RNA yields sufficient for downstream analysis. Summarizing the key points in relation to EV isolation, isolating particles from biological fluids using methods based on hydrodynamic size, density, or charge result in heterogeneous isolates. Because of the possibility of co-isolation of both membrane-bound and non-membranous particles, this may hinder robust characterization of EV, their contents, and their biological activity. The isolation of tissue-specific or enriched subpopulations of EVs really can't be achieved using methods that rely on characteristics that are common to or shared by extracellular particles in EV. It's just not possible. Isolation methods that are not based on universally common features are required to better define and characterize the role of subpopulations of extracellular vesicles, methods such as targeting vessel surface epitopes. In the last 10 to 15 minutes of this presentation, I want to specifically focus on work that we have been doing to develop EV-based diagnostics. And really the first question that needs to be answered is, what's the advantage in measuring EV-associated biomarkers and developing them as diagnostics, as opposed to simply measuring the biomarkers directly in plasma? There is evidence in the literature that EV-associated biomarkers are more sensitive and specific than measuring the analyte directly in plasma. This study by Yokoyama in 2017 measured chorioembryonic antigen, CEA, to classify colorectal cancer in serum samples. When measured directly from serum, the area under the ROC curve was 0.86. When measured in EVs isolated from the same serum, the area under the curve was 0.94. So measuring EV-associated antigens increased the diagnostic performance of the biomarker, increasing sensitivity by 13%, specificity by 38%, and increased overall accuracy by 34%. We see similar results in-house looking at Alzheimer's disease biomarkers in plasma or in EVs isolated from those plasmas. And here we're showing the results for p-tau217, TDP43, and neurofilament L measured both in plasma and in the extracellular vesicle fraction isolated by a product called NeuroNet. And we see a 5 to 8-fold increase in the detection of those proteins. In an ongoing study with Professor Carlos Salomon from the University of Queensland, we have also shown that combining multiple EV-associated markers into a multivariate algorithm outperforms the FDA-approved biomarker CA125 for the classification of ovarian cancer. Interestingly, in this study, the initial work was done using size exclusion chromatography for isolating extracellular vesicles. It has been successfully transferred to ExoNet to improve capacity and to allow the identification of additional biomarkers. In June 2019, the FDA granted breakthrough status to the first EV-based liquid biopsy test. The test was developed by Exosome Diagnostics for prostate cancer, and it measures 3 mRNA transcripts isolated from EVs from urine. The information is combined into a multivariate algorithm and offered as a laboratory-developed test in a CLIA laboratory, while they are gathering additional information to make a full application to the FDA. The breakthrough status means that the application will be fast-tracked. Since then, other EV-based diagnostic tests are being developed, including Mercy Halo ovarian cancer screening test that is measuring EV-associated proteins in plasma. Both the Exosome Diagnostic test and the Mercy test have received FDA breakthrough designation. Other tests for prostate cancer, lung cancer, and kidney cancer are also being developed. As I mentioned previously, we also are developing an EV-based ovarian cancer screening test in collaboration with the University of Queensland. In terms of our path to market, we have completed biomarker validation studies and are now moving on to analytical and clinical validation, so real-world clinical samples. Our plan is to make an investigational device exemption application to the FDA in 2026. This will allow the test to be used in clinical studies, and we'll be seeking approval, hopefully, for that test in 2029. The way the ExoOC ovarian cancer test is run is that we are isolating extracellular vesicles from 0.5 ml of plasma using ExoNet. The samples are run on a Kingfisher platform in a 96-well format, so we can easily run 500 samples a day. The EV-associated peptides that are captured using ExoNet are submitted to mass spectrometry analysis, initially to identify candidate protein biomarkers, candidate peptide biomarkers. Subsequently, having evaluated many candidates, a multiple reaction monitoring assay was established on mass spectrometry that specifically targets 57 different proteins. The information from the mass spectrometer is then fed into a multivariate algorithm and a multivariate index assay developed. And here we see the results of the MRM assay expressed as receiver operator characteristic curves for the 57 peptides to correctly classify case and control samples. I've also identified cancer biomarkers that turn up, so CRP, CA125, CA15-3, and CD9 is also present. Based on the MRM data, we are able to generate cross-validated multivariate algorithms that perform extremely well as classification models for all-stage ovarian cancer. Here we can see the receiver operating characteristic curve, where the area under the curve is 0.98 and has a sensitivity of 91% at a specificity of 98%. This is extremely encouraging. We see little decline in the performance of the algorithm, even with stage 1 ovarian cancer, although we have low numbers in that group at this point in time. The next phase of the study will involve the analysis of real-world clinical samples to further confirm the performance of this particular algorithm. From our own work and the work of others, the translation of EV-associated biomarkers into clinically useful diagnostics represents a feasible and realistic opportunity. The identification of subpopulations or even disease-specific subpopulations of extracellular vesicles may significantly improve diagnostic performance. The use of magnetic beads to isolate extracellular vesicles affords the opportunity to fully automate the process, thus achieving the capacity necessary for delivery of a diagnostic in a clinical pathology laboratory. Given the focus of the meeting, it would be remiss of me if I didn't mention NeuroNet. NeuroNet is an EV isolation tool. It's for isolating brain-derived extracellular vesicles from biofluids. And I'd just like to show you a few slides on the data that we have to date. NeuroNet is constructed the same way as ExoNet. It's built on a paramagnetic core, upon which is fabricated a three-dimensional matrix of affinity ligands. In this case, 9 monoclonal antibodies to surface proteins that are found on oligodendrocytes, microglia, and astrocytes. It contains no antibodies to the common tetraspanin proteins that are expressed on many extracellular vesicles. It behaves quite differently to ExoNet, isolating and enriching a subpopulation of tissue or cell-specific extracellular vesicles. As part of the analytical validation of NeuroNet, we have compared NeuroNet performance to size exclusion chromatography. Here we've used 0.5 ml of plasma for NeuroNet, 1 ml for size exclusion chromatography, and sought to identify oligodendrocyte and microglia markers using Western blot. And as you can see from the images, we're able to visualize a strong signal using NeuroNet, which is absent from the seismic exclusion chromatography extracts. As part of our ongoing clinical validation of NeuroNet, we sought to identify known markers of Alzheimer's disease in extracellular vesicles isolated from plasma using NeuroNet. The data we show here are Western blots for phospho-tau 181 and phospho-tau 217 in Alzheimer's disease and in healthy controls, and we see clear distinction between case and control. Those samples were also subjected to proteomic analysis, mass spectrometry analysis, and we were able to identify more than 200 differentially expressed proteins. 47 of those were unique to Alzheimer's disease. We've now completed this analysis for 100 samples, and the data look extremely promising in terms of identifying meaningful EV-associated biomarkers and the possibility to develop clinically useful diagnostics based on those particular proteins. We have also completed proof of concept studies for Parkinson's disease and see a similar efficacy of NeuroNet in isolating relevant biomarkers. To summarize the key points for NeuroNet, the isolation is not based on commonly shared biophysical features or EV surface proteins. It enriches a subpopulation of EVs that are defined by 9 different epitopes that are preferentially expressed by brain-derived EVs. Proof of concept has been obtained for Alzheimer's disease and Parkinson's disease, and we can identify known biomarkers of those conditions. NeuroNet is not on the market. It's made available on a project basis with key collaborators. So, in conclusion, some final comments. The delivery of clinically actionable EV-based diagnostics is enabled by the identification of disease-associated biomarkers in relevant EV subpopulations. And this is critically related to the isolation methods utilized. Delivery of EV diagnostics requires rapid, reproducible, scalable sample processing that is compatible with a pathology laboratory workflow environment. And finally, it's important to secure early coverage of all R&D activities within an appropriate quality management system. Such a system mitigates many of the translational barriers to moving EV biomarkers into clinical applications. I'll finish there, and I hope that you have found this video presentation of use, and I'm happy to respond to any questions that people have or inquiries that people have via email. Thank you very much, and I wish you well for the conference.